So, thinking about personalized treatment for small cell lung cancer, this is what my lab actually spends a lot of time thinking. So, when we think about personalized medicine for any cancer, there are two main pillars. We think about biomarkers and we think about novel therapies. And this is what we spend most of our times in. So, for the talk today, we're going to think about treatment choices. What are there right now? And we are also think about biomarkers. Are we there yet about selecting patients for these therapies that are currently available? What are some promising biomarkers and what are some of the emerging biomarkers that are either in the lab or in the clinical trials right now? So, as I mentioned, immunotherapy has been here for since 2018, and we do see that a minority of patients derive benefit. However, our effort has been how can we raise the tail of the curve? And what I mean by that is how can we have more patients respond to this frontline therapy? Because we have the most benefit. If we can treat patients up front and if we can get benefit up front, that's always the most beneficial for these patients. So always, I think the goal is to have derive the most benefit from the frontline therapy for patients. So really the question is, how do we raise the tail of the curve? So, well, we've spent a lot of time thinking about these group of drugs that's really cell cycle checkpoints or DNA damage response inhibitors. Studies from our group and others, we have shown that these small cell lung cancer patients with small cell lung cancer, they have a vulnerability, they have a lot of genomic instability because these patients have a loss of two very important genes called P53 and RB1. And they have a vulnerability in these pathways or in these genes that control the cell cycle and the repair of DNA. And the G2M kinases, or the kinases that control the cell cycle, like PARP, CHEK1, ATM, and ATR, they are expressed in a higher percentage in small cell lung cancer, and they are also a vulnerability in small cell lung cancer. So we have published quite extensively and showing that several inhibitors targeting these proteins, like CHEK1 inhibitor, V1 inhibitor, PARP inhibitor, are important drugs targeting small cell lung cancer. And there are several trials that have been done in this field. We have had mixed responses for these drugs. We have seen many failures here, and I'll talk about, combining regimens of DNA damage response with potential immunotherapy. But the point is, there is a vulnerability. DNA damage response inhibitors have moved to phase 1 to 3 trials for patients with small cell lung cancer. This is a study that was led by Anish Thomas, a group at NIH, and they've combined an ATR inhibitor, which is again a DNA damage response protein controlling the cell cycle. So when they have combined ATR inhibitor with berzosertib, so ATR inhibitor berzosertib with topotecan, what they have seen, and they have collected pre-treatment patients with 13 samples and also post-treatment 4 samples. And in collaboration with Anish, when we have analyzed the sample, something really interesting came up. We have seen that berzosertib, the ATR inhibitor, when combined with topotecan, we are seeing upregulation of many, many pathways that are involved in immune regulation. And what that led us to see in the lab is that many of the pathways that are controlling the innate immune response pathway called the CGAS-STING pathway are actually activated when you treat small cell lung cancer in the lab with ATR inhibitor or other DNA damage response inhibitors. And this pathway is called the CGAS-STING pathway. And this is something we have done in the preclinical models where we have shown that combining a DDR or a DNA damage response inhibitor with immunotherapy could give very good response in the preclinical models for small cell lung cancer. We have published extensively in the field where we have shown that when you target small cell lung cancer preclinical models, with a DNA damage response inhibitor like ATR, V1, PARP, or CHEK1, what it does, it launches a multimodal immune activation cascade, which activates the innate immune response pathway called the STING pathway. And it activates the HLA, or the antigen presentation machinery. It also recruits pro-inflammatory chemokines, and you can see some of the names here with CCL5 and CXCL10. It also recruits CD8 positive T cells, so it activates the anti-tumor immunity, and that leads to the decrease in the tumors in the preclinical models. And since then, these drugs have also been taken into clinical trials for patients. And these are some of the trials where the DNA damage response inhibitors are being combined with immunotherapy for patients with small cell lung cancer. Another method of actually augmenting the response of immunotherapy is using epigenetic modifiers. So epigenetic modifiers like EZH1 and 2 and LSD1, we and others have shown that the antigen presentation machinery in small cell lung cancer is actually suppressed by these epigenetic modifiers like LSD1 and EZH1 and 2. And when you combine inhibitors targeting LSD1 or EZH1 and 2, you can reactivate the immune system, or you can reactivate the expression of MHC1. So now there are several trials. I'm showing you an example with an LSD1 inhibitor with nivolumab, which is an anti-PD-1 antibody. I'm showing you one of the examples, but there are other trials that are ongoing that are combining epigenetic modifiers with immunotherapy to augment the response to immunotherapy. So the point is, there are several mechanisms by which we can combine immunotherapy with these agents to augment the response to immunotherapy. And now there are these amazing new classes of drugs that are really shifting the needle for patients with small cell lung cancer. These are the T cell engagers, and that are combining. So they are really, and the most famous one is the tarlatamab, which is a DLL3 targeted T cell engager that is targeting DLL3 on the cancer cells and CD3 on the tumor cells. And they're actually bringing the immune cell to the cancer cell. So it's almost a meet cute of the cancer cell to the immune cell. And it is bypassing this immunosuppressive phenotype of small cell lung cancer. We have seen amazing data in the second-line setting with tarlatamab, in the maintenance setting for tarlatamab, and more recently in the frontline setting with tarlatamab. So it's actually changing the treatment landscape with these T-cell engagers. And I think we are really seeing a shift in the needles, and this will be a new class of immunotherapy for small cell lung cancer. And there are other, antibody-drug conjugates, and there are several right now. There are antibody-drug conjugates against DLL3, anti-CD56, TROP2, CD276, CES6. There are radioconjugates, and the list goes on. And there are, I'm giving some of the examples here. I think there are several trials that are showing really good data. But again, we do have to see those data mature out, and we still have to see whether we see these agents actually helping the patients with small cell lung cancer. So again, but these are the new classes of drugs where we are seeing these patients actually respond in the second-line setting for small cell lung cancer. So the conclusions for this part is that there is a cohort of patients with small cell lung cancer, small but real, that have durable benefit with immunotherapy. The strategies are to improve the response to immunotherapy, and we need to define who are these patients who have durable benefit. MHC repression is one of the ways how they bypassed why these majority of the patients, they don't have response to immunotherapy. And one of the mechanisms by which we can bypass this MHC suppression is through MHC independent mechanisms through these bispecific T-cell engagers. and also through combining them with several agents like DNA damage response inhibitors, and also through epigenetic modifiers like inhibitors to EZH2 and LSD1.